I'm going to introduce work package 3 just for you for orientation. We are building now this program through several work packages, but perhaps the most important thing in this presentation and generally in these 2 days is to promote more of us to think together about what's the best thing for us to do, what are the optimal technologies to invest in, and work package 3 is exactly on this. So we try to map, define, and refine 1D, 2D, 3D, and 4D. I'll explain what do we mean by that. Multi-omics technology, and try to understand which of them and how they can scale to tackle medical applications. And in particular, this multi-omics work package is within a broader context that you'll understand probably today and tomorrow, which is trying to understand disease as something we can profile using new technologies, but then deeply interacting with computational models and experimental models that allow us to close loops and understand more than we can do today. So mostly I'm going to try to motivate and explain and exemplify what we believe can be done in the coming years and why do we believe so. And I want to start with this very blunt question. So why do we actually need single-cell multi-omics for medicine? Because we are reading in the newspapers that medical big data is already there, so it seems like the problem is going to be solved by Google. And the fact is we think that it's really not the case. So here's one example we are working on, which involves a very large cohort of Israeli patients, almost 5 million at the moment. And we are collecting, together with Clalit Healthcare Services, all of their medical records for almost 20 years of history. And it's amazing to see that all the data, so we are talking about 5 million lives of people, just think about it, okay? Can be summarized in 120 gigabytes, that's it, okay? Can be put on a memory stick. That's all of us in this room multiplied by maybe 100,000 and one memory stick. So you realize that the data we have today, the data that the system is currently collecting on patients is simply not even approaching the level of complexity we need in order to do serious medicine. In particular, the coverage we have on patients in current medical systems is really strictly limited and is mostly focused on people that are already actively have some kind of disease. Here is actual data showing you how much coverage we have on individuals that are generally healthy between 45 to 65 years of age, and only about 15% of them are having interactions with the system that are at an annual rate, so meaning they have 15 data points over the last 18 years or so. So most of the data is extremely sparse. For most of the patients, we simply do not know anything. But of course, we can play we can do a mind experiment and just focus on those patients for which we have data and ask, what can we do with them, with the type of data that the system is currently collecting, with the type of data that digital health is suggesting to work on. And it turned out that you can do very serious work of organizing all this data, and we and others are heavily involved in that, and you can start and build predictive tools. So you can take those medical histories and train machine learning classifiers, and here are some examples of some of the predictors you can get on all kinds of disease. There are, let's say, something like 80 different diseases you can build predictors for. And you are very proud in yourself, and you say, aha, I've done something great. But then you go to the physician, and he will tell you, okay, so you have predicted an elevated risk for a certain disease. Now what? Now what? Okay, what can I do with it? What can I offer my patient to do with that? If I can only tell him that he has an elevated risk of some disease, but I cannot do anything about it. And to begin with, this was rare and remained rare. So that's a very acute problem when you think about current digital health and how it is going to be actually become applicable. So the problem is that predictive power doesn't imply ability to treat, doesn't imply ability to make a real impact on medicine. We want in lifetime to intercept disease or to be able to try and understand what is happening and also be able to offer strategies for stopping or slowing down disease processes. So in lifetime, we need technologies that allow us to infer cause and effect, to go into the mechanism of the disease, and we want technologies that allow us to develop new targeted therapies, not just profiling the patients and predicting. In other words, digital health is really today like weather, okay? Everybody is talking about it, but nobody does anything really, okay? So people get VC money, okay? They can get people to invest money in the companies, but they are really not capable of curing patients using digital health at the moment, and we really believe it can be changed. Why it can be changed? Because of dramatic technological developments over the recent few years that many partners in Lifetime and others in the world have pioneered, which allow us to sample molecular processes within patients at unprecedented resolution. So it all started with sequencing technology and with genomics about 20 years ago, with this boom of technological boom. But this rapidly scaled first to be able to sequence germline genetics for many, many individuals, but then over the last 10 years or so, quickly allowing us to profile many other molecular properties of biological systems, including transcriptional states, epigenetic of various types and kinds. This is becoming really powerful. What you see in the timeline here show you how these technologies are scaling, and we actually color-coded what we believe as the threshold that allow us to start and think about medical application. And some of the single-cell technologies are really there in terms, at least, of scaling, in particular, transcriptional analysis. But just being able to scale is probably not enough, right? So we need to understand together how can we take those scaling capabilities and actually do something meaningful with them, find the right applications, find the right paradigms that allow us to use those technologies. In particular, in Work Package 3, we are interested in several layers of single-cell multi-omics. We are starting from one-dimensional techniques. By one-dimensional, we think about the genomic dimension. We can think about two-dimensional techniques that looks like pathological slides and take snapshots of tissues in a certain frozen section, for example. We can think of three-dimensional technologies that allow us to look at overall tissue architecture or go deep into cells and ask questions about how their intracellular and even internuclear structures are looking like, and go into the mechanisms using these approaches. And we can think about the temporal dimension, which is critical in any kind of disease, and extend it further and generate data sets that are really 4D. So I want to end this very quick presentation by giving you two examples and motivate you to think about more. And these are examples of disease that seem to be amenable to those kinds of approach even today. The first one is acute myeloid leukemia. Here you show incidence plots from actual data showing you that this is a very rare disease. So in relevant ages, it's going to happen at 0.01% annually per individual. You see data for males and females. On the other hand, it's a very deadly disease, so getting it is a very poor prognosis. You could do bone marrow transplantation, but that's it. So you can now look at the retrospective data in electronic health record and ask questions about what is happening before disease onset, because once you identify the disease, you can go back in time and ask what is happening. And you can actually, in a paper with Liran Shlush, we were able to predict or to generate a risk stratifier or a predictor that tells you that a certain individual have a 30-fold increased risk of getting an AML, which sounds a lot, right? It sounds a lot, but it's not really a lot because the absolute risk for this individual that is at high risk to AML is still very, very low, and we cannot do anything about it. So what next? So in this particular study, Liran was able to use very deep profiling of somatic genetics to identify actual mutations that are driving disease progression. In that case, those mutations are not only providing us with a better predictor, but they are also giving us certain therapeutic avenues because those mutations, some of them at least, can be targeted. So that's a wonderful example of a disease that is looming there for a while, for many years, okay? You can track and identify it using the new technologies, and once you do it using single-cell multi-omics, or any kind of deep genetics approaches, you can actually come up with therapeutic avenues. A second example is multiple myeloma. Here you see the incidence plots. For this disease, it is a bit more prevalent, about 0.1% for aging people. Again, male and female, not the same. It is also coming with a hefty price tag in terms of survival, although people are being treated and managed for using several lines of drugs in this disease. And here, you can also use the EHR and develop risk stratifiers. So this disease, to begin with, is a very progressive disease where people are at risk, but even the best current medical practice only tell you that you have a certain elevated risk. For example, increasing your chance of getting the disease from this background rate to something like 1% annually. So still a very low rate. So many people are diagnosed as being having something that is called MGUS, and they are at constant risk for progressing into multiple myeloma. Not much can be done about that. Or actually nothing can be done about it except for watchful waiting. So you can use the EHR to further improve your estimation of the risk. So quite significantly, let's say even fourfold. But is this enough? Again, what can we do about it? That's not good enough. So Ido Amit from the Weizmann, together with us and others, and a big consortium of hematologists in Israel, have actually went all the way to apply single-cell RNA-seq on plasma from such individuals, generating those frightening clouds where each point is a cell, and the color indicate if the patient is normal or cancer or myeloma or in some predisposition to cancer, and was actually able to show not only that certain myeloma cases can be very well identified, and we know now which genes are driving the disease in each of them and what kind of heterogeneity is happening in them. More than that, in patients that are currently considered as at risk to myeloma, you can find small cell populations years before disease onset that are actually already constituting the disease clone. In that case, you have an actionable target because myeloma is being treated, okay? So if you go forward with this approach, you can imagine new types of management protocols for those individuals, dramatically increased risk stratification for this entire disease. So these are cases that are identified by the fact that biomaterial is relatively accessible. It's the blood, okay? A disease is relatively well characterized, and we have a good early opportunity to apply our technologies to these approaches. But in general, we need to think about many additional techniques, many additional applications that are going along the same line. Some of them will be, of course, more difficult, and use these exciting capabilities of AI and machine learning, which are really, really there and are really meaningful, but provide them with data, let's say, that is AI-worthy. Not data that doesn't really allow you to do much more than publish papers. So this is what we really want to do in Lifetime. We want to bring those two concepts together, and Work Package 3 is about finding the right technologies that can feed AI with the best data. So we are actually inviting you, participants here and in general in the community, to think hard, to come up with use cases and ideas of diseases that are characterized by the right features that will allow you to apply this scheme, where sample is available, where disease is prevalent enough, where the mechanisms, you can imagine what they are, where biomaterial is single-cell ready, so you can actually perform the protocol or do some adaptation and perform them, where there is a chance for potential therapy. So I think I'm ending up here. There is some time for maybe 1 or 2 questions. Thank you very much. Questions?
Thank you very much, Amos.
You continue. Otmar.
Okay.
Well, Amos, what is the idea about generating data in the long run from the 85% of individuals you don't have sort of clinical or medical records?
So of course, it is really interesting, important, and it's going to happen for sure, okay? To generate regularly tracking of clinical indicators on essentially all individuals, including healthy individuals. This is something that is happening. I think our main point is that it's not going to be enough. It's going to be essential. The fact that we now have coverage on a significant population allows us to start and design experiments and go deep into the molecular mechanisms of those progressive diseases in ways we couldn't think about before. But just relying on like complete blood count and standard chemistry, okay, or even continuous monitoring of saturation and other wearable sensors to make predictions on diseases that involve a whole slew of different mechanisms. This, we believe, can be greatly improved if we incorporate the mechanistic profiling schemes into the system. And we have now the opportunity to do it. It wasn't really possible before, but if you think about it, why do we measure patients using the kind of indicators we currently use, okay? Why are we doing complete blood counts and just the chemistry we do today? This is because it is relatively cheap and because this was probably the technology that was available in the 50s. It's not, why should we do AI with these indicators and not with the indicators that are the right indicators for our time? Doesn't make sense.
I want to say, so Amos was very good. There's actually a few minutes for questions, so don't be shy. And actually, there are 2 microphone stands, so don't hesitate to go to the stand and ask questions.
Nice presentation. You talk about disease, you talk about RNA, completely missing the protein level. The proteins are the interaction partners. Proteins have PTMs. PTMs are quite important, like sugars. Missing the single proteomics part in your presentation. What's your ideas behind?
So that's an excellent comment, okay? And it's actually part of the reason for doing this meeting and for building up our community. We were definitely biased to RNA and epigenetics when we were starting to think about those problems, but we shouldn't be, okay? And the only reason for not including, let's say, proteomics in some of those studies is that, for us at least, or for some of us, it's more difficult to do. But for sure, technologies for single-cell proteomics, or in general, any technologies that can target those molecules are extremely important and useful. In some disease, they will make the difference, for sure. Point well taken.
Perhaps just a comment in that direction. It's obvious that the presentation have to focus on particular aspect, and it's just an illustration with some examples, but obviously there's a lot more that we can do. So I think this is inspiring, and if you have suggestion for how, with other approaches, we can incorporate these additional parameters, it will be even stronger.
And given that, I want to also comment that the fact that I picked up those 2 particular examples on hematological disorders doesn't mean that Lifetime is intending to focus on those. By all means, there is no decision like that, okay? We are completely open to any kind of application. These were meant as examples. And remember, what I'm trying to promote here is a practical plan to think about things that we can do now. That's the message.
A question just to say for the gentleman that single-cell protein quantification has, of course, started to be developed. And in fact, it is already possible to quantify a few hundred antibodies binding to individual cells by these kinds of methods. So I think it's just a question of time and the people coming together to do this. But there was a question over there, and if you want to answer, you can, of course. But maybe first, the gentleman in the back.
Thank you very much. Thank you very much for the very nice lecture. These were 2 hematologic examples, very nice examples. But I would like to ask how to generate single-cell profiles from very complicated cells. Like which structure is very, very complicated, like for example neurons or Schwann cells. So you basically, if you homogenize the brain or get out the cells, then you're missing axons, dendrites. You miss whatever myelin sheets. There is plenty of mRNA inside there.
Absolutely right, and there are wonderful examples today of scientists, even in this consortium, that are actually actively profiling such cells with great success. The application to disease is something that we are starting to see. And by all means, the fact that I gave 2 examples on blood is just because this is the easiest to start with, okay? where sample collection is the easier, but our collective challenge is to be able to apply these techniques to many, many other systems, not only neurons. Of course, in cancer, in solid tumors, you see huge amount of progress. This is perhaps also one of the only fields in which precision medicine is actually does cure patient. So these are just examples, and we are obliged to find many, many others. Please.
Thank you. Just a point. First of all, a really well done opening to this conference. Recently we've been able to look at so many different types of receptors on the surface of a cell. And when we begin to do that beyond where we've ever seen before, the complexity of that receptor proteome is really dynamic, even in normals. So before we jump into diseases, a good question might be, do we have enough information about normals?
Okay, so that's also an excellent question. It also relates to Nicolas and Genevieve's comments about relationships with the HCA. So we are actively participating in a big effort to try and map normals and understand what is the reference, which we greatly missed for many years. So this is by all means not finished, okay, but it's an ongoing process that is keep improving, and we already have some preliminary atlases of some of the important tissues out there. So I think Lifetime is relying on the fact that this progress is going to continue. And by the way, the HCO is also relying on the fact that initiatives such as ours are continuously feedbacking with the HCA. So, and more specifically with respect to receptors or the ability to profile cells using techniques that are not necessarily genomics, this is also completely on the table. And by the way, part of the breakout sessions discussion tomorrow will try to promote people to express their opinions about it. What is the right thing to do? Should we quickly scale and do more patients with fewer markers? Should we start by going as deep as possible and as multi-layered as possible for fewer patients? These are all open questions for which we have no answers ready. Okay, we need to work on it together.